So we continue with the fungi part. So now we see the role of the fungi, useful role of fungi. First is antibiotic. An antibiotic is a chemical substance produced by living microorganism which can stop the growth or kill or other microorganisms. The first antibiotic was penicillin, produced by a mold. You have read it about in the earlier part. To refresh your knowledge, it is criteria of good antibiotic. It should be able to kill a variety of disease-producing microorganisms. Broad spectrum antibiotic. It should not produce undesirable side effects. It should not kill normal bacteria of the host. This was already discussed in the earlier videos. Other useful use of antibiotic is as food preservatives, especially for fresh meat and fish, for treating animal feed, and for controlling plant pathogens. Now we see the fungi role in industry. Fungi have a very extensive use in industry. Here we will talk about the large number of practices that are quite old and traditional and some that are rather recent. First is wine, an alcoholic beverage. Wine and other alcoholic beverages like beer are products of the process called fermentation. Fermentation is a process in which the microorganism, yeast and bacteria break down carbohydrates into simpler products, ethanol or lactic acid, in the absence of oxygen. So it is seen that certain bacteria and the yeast is going to respire anaerobically in these particular kind of juices, and these are going to ferment it. That means it is going to cause formation of alcohol and going to add alcohol to that particular thing, causing converting those juices into alcoholic beverages. The process is called as fermentation. The source used for making wine is grape, and for beer is barley maltose. And microorganisms employed is yeast. The biochemical reaction is as follows: that C6H12O6 will give you C2H5OH plus CO2. Grapes are crushed and fermented in large vessels. Yeast cells, either naturally present on the grapes or added, carry out the process. Ethyl alcohol is produced at the concentration of about 12%, and this concentration starts killing yeast cells. What is left is wine. Spirits such as whiskey and gin, which have much higher alcohol content, are obtained by distillation of fermented mixture. Wines contain variable amount of water, alcohol, carbon dioxide, sugar, and a few other substances. So it is seen that wine is going to be produced by the grape juice. So the grape juice is taken. Now the grapes will be having certain yeast in them, or there will be some added yeast to this particular grape juice. On adding the yeast, the fermentation of the grape juice will start, and there will be a production of alcohol inside the juice. Till the percentage of the alcohol becomes 12%, the yeast is able to survive inside. But as soon as the alcohol content increases by more than 12%, the yeast is going to get killed. So that is why what is going to remain after that will be only the alcohol, because of that is wine, and because the yeast has already been killed. In the other cases, like formation of gin and whiskey and all, where the percentage of the alcohol is very high, the high percentage of alcohol is produced, or it is formed by distillation of these particular juices. So this distillation, after the production of the alcohol by the yeast, it is then distillation is being done, and accordingly the alcohol content is increased. So that's why wine contains variable amount of water, alcohol, carbon dioxide, sugar, and a few other substances. Characteristic of wine: wine is quickly absorbed into the body. So yes, wine is in a small quantity. Yes, wine is good for health because it is a good appetizer. But of course, you don't know what is the good quantity, and that's why it leads to problems. In a very mild quantity, it may be a stimulant, but when consumed in large quantity, it is harmful in many ways. Alcoholics may develop body ailments, particularly liver cirrhosis. Now, the damaging of the liver is the most serious problem which can happen due to the regular intake of alcohol. So alcohol is definitely very bad for the health. Only in very small quantity it can act as a stimulant and help be a appetizer. It will be good for your digestive system, but in a very small quantity. But normally people are not able to get that take it in a small quantity, and they lead to problems later on. The next is baking, that is bread making. The next industrial use of fungi will be bread making, yeast. In baking, a mixture of flour, atta or maida, some fat, salt and water is required to make the dough. The starch of the flour gets converted into sugar. When yeast is added, it ferments and sugar and produces carbon dioxide, and the dough rises, leaving to about 3 times its original volume. When the dough is baked, the gas bubble expand and give the bread a slight spongy texture. See, what in case of the bread making, they take the dough is prepared by the atta and everything, so maida or something, and then the flour and everything is being mixed up and they form into a dough. Now that dough is going to be set aside with the yeast present inside. The yeast present inside is going to start producing carbon dioxide due to respiration. This carbon dioxide which is produced will automatically increase the size. They will get entrapped into the pores of this particular atta and that dough, and then when it is put into the oven, at that time this carbon dioxide will try to release out, and जहाँ जहाँ पे वो था वहाँ पे वो expansion होगा, and that's why the bread gets the texture of that bubbly that porous kind of a material is going to be. So the sponginess of the bread is going to be achieved due to the pores which are formed by the carbon dioxide present inside that particular dough. The next is cheese. Cheese is valuable food with high quantities of fats and proteins along with calcium, phosphorus, vitamin A and some quantities of vitamin B. People have been eating cheese for centuries. It has been made from milk. The manufacturing of cheese include the following major steps. So cheese, of course, we know very well. We love that thing, right? So that is the high quantity of fats and proteins are there in it, with a good amount of calcium, phosphorus, and vitamin A. It is basically we normally should not eat too much cheese. Basically, the fat content. The fat content is very high in cheese. That's the main problem with us. So it's because when we eat certain kind of food like pizzas and pastas and all, it has got so much of cheese in it, which is going to create a lot of kind of fats. The fat intake becomes too high. That's why it is not advisable to have too much of cheese. But otherwise, it has got good amount of nutrients in it. So anyway, so we are going to see the major steps involved in the manufacturing of the cheese. So first is the curdling of the milk. So first thing what they do is they convert the milk into a curd. So by addition of lactic acid bacteria, lactobacillus, curd produced is separated from whey. Liquid remained after curdling. So what they do is that they are going to convert the milk into curd, and then it is going to be tied up so that the liquid part is being removed and the solid part is extracted. So the solid part, which is the going to be whey part, which is the one which is going to be used for making the cheese. Curd is processed to remove moisture. After this stage, it is called as cottage cheese. So cottage cheese is nothing but paneer. So once that particular whey which is taken, if उसमें से पानी पूरा extract कर दिया, तो what we are going to get is nothing but the paneer, that is cottage cheese. Salting, this further removes moisture and prevents growth of undesirable microorganisms. So then what they do is that they do salting. Salting करने से क्या होगा? See, normally paneer cannot remain for a longer period of time. It will get bad. But so to prevent it from decaying, we are adding salt to it. So we are salting the paneer. So by adding the salt, we are removing the moisture and also preventing the growth of undesirable microorganisms. And finally, we are ripening it. So ripening, the curd is kept for suitable temperature and humidity. The microorganism added along with the lactic acid bacteria at step 1 imparts particular flavors to the cheese. So accordingly, that flavored cheese will be formed. The flavor of the cheese is going to be got out, because otherwise paneer normally is almost like very soft taste. The taste is not a negligible taste is there for paneer, or flat taste. Whereas cheese is prominent taste, so that prominent flavoring of the cheese is because of the addition of the lactic acid, which is going to be bacteria which are added in the first step of curdling of the milk. Next is mushroom cultivation. Mushrooms are of various shapes and sizes. Many have a cap and a stalk, but in some variety the stalk is absent. There are a large number of mushroom species which grow wild in nature. Not all mushrooms are edible. In fact, most are mildly or severely poisonous. The edible ones have been eaten by humans since ancient times. Agaricus bisporus, bisporus is the most common edible mushroom eaten practically all over the world. At present, three kinds of mushroom are widely cultivated in India and abroad. First is white button mushroom, Agaricus bisporus, paddy straw mushroom, Volvaria. Oyster mushroom is Pleurotus. Pleurotus, yeah. Mushroom cultivation is done indoors and hence little land area is required. So mushroom cultivation is done indoors. It is done inside the house. It is not done outside in the field. Okay, it is done inside the house. So inside land. So it is cultivated indoors and hence little land is required. Mushroom can be grown in substrate or compost based on various agricultural waste, which in turn are recycled. Compost decayed organic materials used for fertilizing land. So you do not require soil, you will be requiring compost, that is the manure, or you can say the decaying organic material which is used for fertilizing the land is compost. So they are going to take compost directly, on the compost the growth of mushroom is going to take place. White button mushroom is most popular variety grown in India. Its cultivation involves 5 major steps: composting, spawning, casing, cropping, and harvesting and preservation. Composting. The compost is prepared by mixing of the following in certain proportions: wheat or paddy straw, chicken manure, some organic and inorganic fertilizers. This compost is kept at about 50 degrees for around 1 week. So they are going to prepare a compost that is a mixture of wheat and paddy straws, chicken manure, the one which is given to the chicken for food for chicken manure, some organic and inorganic fertilizers. So these all are going to be formed into a compost and is going to be kept at around 50 degrees Celsius for around a week. And then they are going to spread the seeds of mushroom, or the spawning. The mushroom seeds consisting of mycelium. So we have learned this thing, this particular sporangium. So that particular mushroom can be ऐसे sporangiums होते हैं, that is which is forming into mycelium. So these mushroom seeds consisting of mycelium or selected type of mushroom is introduced into the compost and allowed to spread for a couple of days. Then casing, a thin layer of soil is spread over the compost. This is a very important step because it gives support to the mushroom. So of course it will require some support for its growth. That's why there will be some soil which will be spread across this particular layer of the compost after the spawning is done. This soil what we have spread will give it a support for the mushroom. It provides humidity, prevents quick drying of the compost, and helps to regulate temperature. So all these are going to help the compost or to remain intact and help the growth of the mushroom and give it complete nutrition. The next step is cropping and harvesting. So the growth occurs in 3 principal stages. Mycelium, a network of fibrous mass, grow within 2 to 6 weeks. Tiny pinheads are going to be formed later on, and button stage which grows bigger, attaining marketable size. The full grown mushroom are then taken out. So that is how the first वो छोटा सा बनेगा, और उसके बाद में within around 6 weeks time the mycelium will be formed and then it will be start growing into a proper mushroom, button mushroom. Finally, remember this is a kind of a fungus. So what mushroom are you eating is a kind of fungus. So definitely you need to have some kind of preventive preservation which is required. Mushrooms are highly perishable. Their shelf life is increased by a variety of processes. vacuum cooling, giving gamma radiation and storing at 15 degrees Celsius, and freeze drying, that is sublimation of ice from frozen mushroom at a reduced pressure in a solution of citric acid, ascorbic acid and brine, etc. So these are the three processes which are done to mushroom so that it can remain for a longer period of time without any problems. Now, nutritive value of mushroom. Mushrooms are excellent source of vitamins and minerals. They contain a good amount of niacin, pantothenic acid and biotin. So yeah, these are the three kinds of nutritions which are available from mushroom, that is niacin, pantothenic acid and biotin. The vitamins in mushroom are well retained during cooking, canning and dehydration. So that is the main advantage. Normally it is seen that whenever any vegetable is being cooked, it will lose its vitamin value and it will lose its nutritive value also. Whereas in case of mushroom, the advantage is that even while cooking or canning or dehydrating, the nutritive value of that particular mushroom is not lost. So that's the main thing, that the vitamin value is retained during cooking, canning and dehydration. Fresh mushroom contain about 85 to 95% moisture, 3% protein, 4% carbohydrates, 0.3% to 0.4% fats and 1% minerals and vitamins. Caution, never try eating mushroom from the field. They may be poisonous and even fatal. Buy them only from reliable shops which obtain their supplies to the mushroom farms. So this is how the mushrooms was cultivated and is given the nutritive values, and hence this was the end of the chapter bacteria and fungi. So quite a small chapter as such, with not too much to learn. Even the importance of this chapter in the board exam is not very high, so you can just go through them in a proper manner so that you can easily cover this topic, and you've got very simple things over here to learn which can be easily be identified. So that was about the chapter bacteria and fungi. Thank you.